Greetings, ladies and gentlemen. Once again, we are going to be looking at the genetics of the ABO blood system. The genetics of the ABO blood system. The ABO blood system is a classification of the human blood, and this system is primarily made up of what we call the H antigen. The H antigen, which is a precursor of the antigen A and antigen B. The primary precursor is what we call the H antigen. And this H antigen, of course, is a carbohydrate that is found on the red blood cell. And this is what determines if someone will have antigen A, or let me say blood type A or blood type B. This H antigen works in conjunction with an enzyme that is called alpha 1,3N acetyl galactosamine transferase. Alpha 1,3N acetyl galactosamine transferase. It is commonly known as the GTA or A transferase. So what this enzyme does is to add glucose to the H antigen. So H antigen H antigen plus N acetyl galactosamine will result to blood type A, whereas H antigen plus galactose will result to blood type B. So for an individual to have blood type A, it means that the enzyme which I've mentioned, the GTA, adds the H antigen, which is a primary precursor, to the glucose N-acetylgalactosamine. Once this happens, then it results in the formation of a blood type A or antigen A. Then, if the H antigen is added to the galactose, it results to the formation of blood type B. Then for blood type AB is a combination of this and this. Of course, the N-acetylgalactosamine is for A, galactose is for B. So the combination of the two, the addition of the two in addition to the H antigen again is what results to the blood type AB. But there's something that happens differently in the case of blood type O. In the case of blood type O, you discover that the blood type O does not have the GTA enzyme or the A transferase enzyme. And because it does not have it, then the only antigen that remains within this blood type O is the H antigen. There is no antigen A and B in blood type A. However, they have antibodies A and B. We are still going to look at this holistically. And like I mentioned, the primary reason is because the blood type O lacks the GTA enzymes that helps in the incorporation of glucose or carbohydrate molecules into the precursor, which is the H antigen. So, in essence, if we have this, we'll have this, we'll have this, and we'll have this. So this is blood type A, blood type B, blood type AB, blood type O. So this has A antigen on the surface of the red blood cell. Then within the plasma, this person will have antibody B. Then this person has antigen B on the surface of his red blood cell. Then within the plasma, the person will now have antibody A. So you can see the reverse of A, antibody B, is found in blood type A, whereas the surface of the red blood cell contains antigen A. So in this case now, the surface of the red blood cell contains antigen B. Remember, this is a product of the reaction between the H antigen and the GTA enzyme that resulted to this. So for AB, they have AB antigen on the surface of the red blood cell, then no antibody. There's no antibody at all. Then for O, because the O, like I said, lacks the enzyme, the GTA enzyme, so nothing to incorporate the H antigen with any glucose. So it is only left with the H antigen. So in this case now, there is no antigen on the surface of his red blood cell, but it has antibody A and antibody B. Antibody A and antibody B. Don't forget, it's that reaction that brings about whether a blood type will be A or B. So since the enzyme to do the job is not there, so there's no antigen that is formed, and it's peculiar to the blood O type. Okay, so since we have known this now, let's now talk about the let's now look at how a blood transfusion is done or should be done. Let's look at blood transfusion, because it is important you understand that the how do I want to put it now? If blood types, during blood transfusion, if the blood of the donor and the recipient is not compatible, then of course it results to a hemolytic reaction. It results to a blood reaction which is called agglutination, and this agglutination is very deadly. So it is always good to screen the donor's blood to see if it's compatible with the recipient's blood. So in this case now, it is also important to let you know before I begin to look at the table which shows blood types that match with each other. It is important to let us know that in blood transfusion, uh, similar antigens and and antibodies do not coexist in the same host. Similar antigen and antibodies do not coexist in the same host. So let me draw that again. I use that to explain this. Similar antigens and antibodies do not coexist in the same host. Do not coexist in the same host. So, like I said, this is antigen A, then antibody B. This should be antigen B, then antibody A. This is antigen AB, then no antibody, then antibody A and antibody B, no antigen. So in this case, in blood transfusion, there's something that should be carefully watched, and that thing is that corresponding antigens and antibodies do not coexist in a single host. So let's look at what I meant. So this is antigen A. Antigen A corresponding. So corresponding, let me put it down. Corresponding antigens and antibodies do not coexist in the same host. So it means that this blood type now, if this blood type donates blood, or if this person, somebody who has blood type A, donates to blood type B, Agglutination will occur. Why will agglutination occur? Agglutination. Agglutination. Hmm? Agglutination. So why will it occur? It will occur because corresponding antigen, this is antigen A, this is antibody A. So this is corresponding. So this cannot work. This is corresponding. A is corresponding to this antigen A here. So because they are corresponding, they are of the same type, they have the same alphabet, corresponding antigen and antibody, not antigen and antigen, but antigen and antibody. So far they correspond in terms of the alphabet, then you will now know that this blood type is not compatible. But if blood type, let's assume this is another person, having the same blood type B, so blood type B, then antigen A. So you can see that this antigen, this antigen, this is the antigen B, then antibody A. So yes, you discover that in this blood type now, this person can comfortably donate to this person because there is no corresponding antigen and antibody. The problem arises when there's corresponding antigen and antibody. So in this case now, we'll have antigen A, we'll have antigen B, we'll have antibody A. So they are not corresponding. So the B can comfortably donate blood to fellow B. But then, let me use, so that I involve other blood types, let me use a table to describe how this should be done. So if we have plus means safe, plus means very safe, then minus means not allowed. Minus means not allowed. So let's assume that we have this blood type. So let's assume that we have this blood type. This is O, this is A, this is B, this is O. Sorry, this is AB, then O, A, B, AB. So in this case, also don't forget that in the ABO blood system, we have what we call the universal donor and the universal recipient. So blood group O can donate to all the blood types, and that is why blood group O is known as the universal donor. Then the AB can receive from other blood types, and that is why the AB is known as the universal recipient. So if O should donate to O, it is very safe. Then O donating to A is safe. O donating to B is also safe. O donating to AB is also safe. Why? Because O is a universal donor. Then A cannot donate to O because A is not a universal donor, so it's not safe. But A can comfortably donate to another A. Then out of A and B, it is not safe for A to donate to B. Then for A and AB, A can donate to AB and is safe. Not very safe, but safe. Then B cannot donate to O, is not safe. B cannot donate to A, is not safe. B can comfortably donate to B. Then B can donate to AB, is safe. Then AB cannot donate to O, is not safe. AB cannot donate to A, is not safe. AB cannot donate to B is not safe, but AB can donate to AB is very safe. So this is title table showing blood transfusion. Showing blood transfusion. So this shows, as a matter of fact, you should have this when people are in the hospital and they want pints of blood. If you look at this chart, then this chart can give you an overview of the possibilities of donating blood from one person to the other. Okay. Then having known this now I said something. I said A can donate to AB, as you can see. So you can see that the whole of this, the whole of this, O is donating to others. This is the universal donor. Then AB received from others. So AB is a universal recipient. So I say that A can donate to AB, but AB cannot donate to A. Let's see if we can look at that. Why A can donate to AB, but AB cannot donate to A. This is A. This is AB. AB has AB antigen. Then no antibody in the plasma. This one has A antigen. Then antibody B. So the reason A can donate to AB is because there is no corresponding antigen antibody in between A and B. There's no corresponding antigen antibody. So look at this now. This is antigen. As you will have antibody B here. Sorry, as you will have, yes, this is antigen. Yeah, as you will have antibody A, then that means that there is a corresponding antigen and antibody. But because there's no corresponding antigen and antibody, this one can donate to this one and there will be no problem. But so this, from this to this, good can go. But from this to this cannot go. So if AB is donating to A, if AB is donating to A, so this is antigen B, this is antigen B, and is corresponding with antibody B. So you can see that there is a correspondence. There is a corresponding antigen antibody. So this antigen B corresponding with antibody B. So that is why AB